Welcome back. We continue this morning's program with Prof Thomas Südhof. So Prof Südhof will be joining us virtually for his talk. His talk is titled Towards Understanding Synapse Formation in Health and Disease, Implications for Neurodegeneration. The Q&A session will be moderated by Prof Lim Ka Leong. Prof Südhof, please.
All hear me. I wish I could have been there in Singapore in person. It would have been so much more enjoyable, I hope, for everybody, certainly for me. I miss visiting Singapore, where I've been several times before the COVID epidemic. And I look forward to my next visit to this wonderful country. What I want to do today is indicated in the title of my talk, except that when I gave this title, I didn't realize that I only had a short time to speak. And so I need to now change that title. And I will change it to reflect what I'm going to talk about, which is a scientific project that we are currently doing in the lab and that I find particularly interesting, that relates to disease, but I'm not going to have time to tell you the details about this disease. In fact, I want to specifically talk only about a very, very brief introduction to one big area of research, which is synapse formation. We all know that the brain functions in processing information via neural circuits. What you see here is a schematic of a neural circuit, a microcircuit, where three neurons are connected to each other by synapses. So circuits are formed by synapses, and the circuits process information by transferring and changing the information from one neuron to the next at the synapses. The synapses are the fundamental computational unit of the brain that exhibit an amazing speed, precision, and plasticity. There are many properties of circuits that are important for information processing. And one of them, possibly the most important, is the location, identity, and properties of the synapses. What my lab is trying to do is to understand what molecular rules govern the formation and specification of synapses and what happens when these rules and the synapses are disturbed in disease. Our fundamental proposition is that in a synapse, as shown here in this electron micrograph, two cells, two nerve cells usually, form an intercellular junction. And that junction is specialized for transferring and processing information. The idea is that this junction is controlled by transsynaptic adhesion complexes that mediated bidirectional signaling, thereby telling each neuron about their identity and instructing each neurons with regards to the plasticity, release probability, and other properties of the synapses. Many candidate synaptic adhesion molecules have been described that are seen schematically in this slide here. This is not an exhaustive presentation. There's many more, but these arguably are the most important. What you see here on the left side are the presynaptic terminal molecules, on the right side, the postsynaptic. And you can see that on the left side, there are several hub molecules, such as neurexins, that interact with lots of postsynaptic molecules. Although we try to understand all of these molecules, in the end, there's only a very superficial understanding at present. And what I want to do is focus on one particular set of molecules that I'm going to discuss today. And what I want to try to illustrate to you today is how we can use molecular approaches to dissect the molecular logic that governs the transsynaptic specification of synapses. And specifically, what I want to focus on today is neurexins, which are presynaptic molecules that interact, as you can see here, with a wide spectrum of postsynaptic ligands. In fact, more than 50 genes encode ligands for neurexins. I don't want to give you a total view of this family of proteins because the time isn't sufficient. I only have 30 minutes. I only want to tell you about one particular project within that bigger project, which is again part of an even bigger project, namely to understand the molecular logic that dictates synapse formation and specification. So this is what I'm going to tell you about today. I'm going to tell you first about the physiological function, not of neurexins as a whole, but of one particular event in neurexins, namely in neurexin alternative splicing. And then I'm going to tell you about the amazing mechanism that is used to actually mediate this function. So let me start at the beginning, and let me first introduce you in more detail to the presynaptic adhesion molecules called neurexins. What you see here schematically is that these proteins come in two principal flavors, alpha neurexins and beta neurexins. The alpha neurexins are larger versions with many more domains than the beta neurexins, which are shorter versions. Alpha and beta neurexins are encoded by 3 genes, each of which has independent promoters for alpha and beta neurexins. They're localized to synapses, at least in part. Interestingly, thousands of mutations were identified in neurexins in autism, schizophrenia, Tourette syndrome, neurodevelopmental delay, all kinds of neuropsychiatric disorders. And we have, in fact, in studies that I don't have time to discuss today, analyzed in neurons derived from human stem cells and from patients the effect of such mutations, demonstrating that they do impair synapses, which I'm not going to discuss because of limits of time. What struck us when we first identified neurexins was that they have a tremendous degree of alternative splicing. They're among the most alternatively spliced molecules in biology, creating thousands of isoforms that have been validated by biosequencing. And what I'm going to talk about today is one particular splice site called SS4 that is shared by alpha and beta neurexins, as shown here. And finally, as you probably know, we have already, as I already mentioned, doesn't work, excuse me. As I already mentioned, neurexins have many, many ligands. So what I'm going to talk to you about today is, in fact, the isolation, the role of one particular splice site in neurexin splice site 4, a very limited, very restricted question. And I'm going to discuss this with you today as a way to illustrate to you how we try to address this big question by going into detail in one particular molecule, in one particular splice variant, marching from one molecule to the next. And so the starting question of the project that I'm going to discuss with you today is, what does alternative splicing of neurexin at SS4 actually do? And so to address this question, we used a genetic approach. We made mice in which alternative splicing of neurexin 3 is under control of Cre recombinases. These mice were made in such a way that in the knock-in, as this is, without a Cre recombinase, you always get a splice variant with SS4 in. As soon as you add recombinase, you always get SS4 minus. And this way we can control very tightly the splice variant of neurexin 3 that's being expressed. Using this approach, we then analyzed hippocampal neurons in culture from these mice electrophysiologically. What you see here is that when you do this, the neurexin 3 SS4 plus has a decrease in synaptic strength mediated by AMPA-type glutamate receptors, whereas synaptic strength mediated by NMDA receptors is normal, as is the synaptic strength of inhibitory synapses. So this was a surprise because this is a postsynaptic phenotype, and it raised a number of questions that we needed to address. Namely, one is, being a postsynaptic phenotype, is this really a presynaptic effect? Does it act presynaptically, transsynaptically on a postsynaptic neuron? And so in order to address this question, we used an in vivo approach that is illustrated here. We injected stereotactically into the CA1 region of these mice, of the knock-in mice, AAVs that encode either wild-type Cre recombinase or a mutant control Cre recombinase at P21. And then we analyzed these mice by electrophysiology 2 weeks later, recording from the subiculum neurons and stimulating the axons that are formed by CA1 neurons and form synapses on the subiculum neurons. This preparation allows a precise analysis of a presynaptic effect because only presynaptic CA1 neurons are infected. The postsynaptic CA1 neurons are not infected with AAVs, as you can see here in the green color that is created by the AAV infection. And when we did this, we observed the same phenotype as in cultured neurons. We found that the neurexin 3 SS4 plus knock-in caused a decrease in AMPA receptor-mediated glutamate receptor responses, whereas the presynaptic excision of SS4 reversed this phenotype and made it appear to be the same as in wild type. We detected no effect on NMDA receptor-mediated responses. So this result confirmed that presynaptic neurexin 3 alternative splicing at SS4 controls postsynaptic AMPA receptor-mediated responses. And further studies showed that this operates at the level of the receptors themselves. So this constitutes a mechanism by which a presynaptic neuron can tell a postsynaptic neuron how to behave, what properties to have. But it also raised questions. The neurexins are highly homologous. Do the other neurexins perform similar functions? Or do they do something different despite the fact that they're so similar? And so to address this question, we generated and analyzed the same type of mutant mice for neurexins 1 and 2. And when we did this, we got a big surprise in that the neurexin 1 mutant had no effect on AMPA receptor-mediated endocytosis, AMPA receptor-mediated responses. They were exactly the same. But when we now analyzed NMDA receptor-mediated responses, we saw a dramatic increase in synaptic strength. So different from the neurexin 3, where there is a dramatic decrease in AMPA-mediated responses, here there is a dramatic increase in NMDA receptor-mediated responses, suggesting that the neurexin 1 and neurexin 3 have completely different functions, affecting glutamate receptors at the synapse by a transsynaptic mechanism, despite the fact that they are so similar. And finally, neurexin 2 SS4 mutants, to top it off, had no phenotype. They didn't affect either the AMPA or the NMDA receptor responses. So the three neurexins are intrinsically different. And these experiments, together with additional experiments that I don't have time to discuss, led to the conclusion, as shown here, that neurexin 1 SS4 plus upregulates NMDA receptors, whereas the neurexin 3 SS4 plus downregulates AMPA receptors, which represents a mechanism of transsynaptic control contributing to synapse diversity. Presynaptic neurexin 1 and neurexin 3 alternative splicing by this mechanism dictate the postsynaptic glutamate receptor content that is characteristic for any given type of synapse. So you might ask, I hope, how does this work? How is it possible for two different neurexins that are so similar to cause the regulation transsynaptically of two different types of glutamate receptors? To address this question, we have to go back to this diagram which illustrates the ligands of neurexins. And it turns out that there's only one type of ligand that binds exclusively to SS4 plus splice variants of neurexins. And these are cerebellins that are secreted adapter proteins. And these cerebellins come into two different types, 1, 2, 3, and 4, that bind to different postsynaptic receptors. When we analyze cerebellin expression in the subiculum, our target area, we found that only cerebellin 2 is expressed at very high levels, and therefore we used cerebellin 2 knockout mice to test whether they might be responsible, whether they might show a similar phenotype. Now, cerebellins have traditionally been implicated in synapse formation. So we first measured synapse numbers in the cerebellin 2 knockout mice in the subiculum and found no phenotype, no change in synapse density. We then measured synaptic transmission, and gratifyingly, we observed both an increase in AMPA receptor responses and a decrease in NMDA receptor responses, as shown here. So what this means is that the cerebellin 2 knockout represents a mirror image of the combination of neurexin 1 SS4 plus and neurexin 3 SS4 plus effects. Neurexin 1 SS4 plus increases NMDA receptors. If you take away cerebellin in a wild type, you get a decrease, presumably because the endogenous neurexin 1 signal is no longer there. Neurexin 3 SS4 plus suppresses AMPA receptors. If you take away cerebellin, you get an increase of AMPA receptors. Again, presumably the endogenous neurexin 3 signal is no longer there, suggesting that strikingly and unexpectedly, cerebellin 2 mediates or transduces the effect of both neurexin 1 and neurexin 3. So what is the cerebellin 2 receptor on the postsynaptic site? Now, it turns out that only one family of proteins has been shown to bind cerebellin 2 postsynaptically, and that is a family of protein called the GLUD1 and GLUD2. And these are remarkable because GLUD1 and GLUD2 are actually homologs of AMPA and NMDA receptors, which are also homologous to each other. So we tested whether GLUD1 or GLUD2 might be the target of this manipulation. We deleted GLUD1 using CRISPR approaches. And as you can see here in culture again, with GLUD1 knockouts, there is no change in synapse numbers. But when you measure synaptic function, you get a phenocopy of the cerebellin 2 deletion, in that there's an increase in AMPA receptor responses and a decrease in NMDA receptor responses, suggesting that the cerebellin 2 effect is mediated via GLUD1. And in vivo experiments showed the same results. Moreover, double mutants demonstrated that GLUD1 functions downstream of neurexins and cerebellins, suggesting that this following pathway operates, whereby neurexin 1 and neurexin 3, SS4 plus, regulate differentially NMDA and AMPA receptors in opposite directions. And this effect is mediated by the same transduction machinery on the postsynaptic side, raising the important question of how on earth can the same postsynaptic GLUT1 molecule transduce distinct neurexin sequences, distinct neurexin signals. And so the hypothesis that we came up with is that GLUT1 transduces distinct neurexin signals via different cytoplasmic effectors, that it basically differentially has different modules for the different neurexins to mediate their effects. And to test this, we probed effects of mutations in the cytoplasmic GLUT1 sequences. We identified 5 conserved sequences in the cytoplasmic tails of GLUT1, and we mutated these 5 sequences, which are shown schematically here, to exactly the same target sequence, except for the C-terminal PDZ domain binding motif, where we exchanged it for a different type. And then we tested rescue. And what you see here on the left is the AMPA receptor-mediated responses. You can see that the knockout, as shown before, dramatically increases the responses. I've shown you this before. What you can also see is that only 1 out of all these mutants did not rescue this phenotype, suggesting that there was one particular sequence that was essential for the AMPA receptor-mediated regulation by GLUT1. When we did the same experiment for the NMDA receptor-mediated responses, you can again see the suppression by the knockout, and you can see that again, only 1 mutant did not rescue. The other ones all rescued. But the mutant that rescued the NMDA receptor-mediated response was different from the one that rescued the AMPA receptor-mediated responses, demonstrating that it's different sequences in GLUT1 that mediated the 2 different regulatory effects. In other words, the mechanisms are, as you would expect, different for AMPA and NMDA receptors, leading us to this model here, whereby the GLUT1 cerebellin 2 complex regulates NMDA receptors on the one hand and AMPA receptors on the other hand, depending on whether it's presynaptically neurexin 3 or neurexin 1. Now, in my final sets of experiments, I want to explore further how this might work. As I mentioned already, GLUT1 is structurally similar to AMPA and NMDA receptors, which operate by a mechanism whereby ligand binding to the extracellular sequences causes a conformational change in the transmembrane regions that then opens an ion channel. So the question then arises whether GLUT1 operates by a similar conformational change in the transmembrane domain like AMPA and NMDA receptors, whether that is conserved or whether they have a different mechanism. And so to test this, we simply took away all transmembrane regions and, in fact, the transduction domain as well. And we made a simple variant of GLUT1 that is shown here schematically, where we change the transmembrane regions, the 3 transmembrane regions, and convert them into a single transmembrane region that we get from CD4. We also took away one of the extracellular domains so that the only sequences that we have are the N-terminal cerebellum binding sequence and the C-terminal cytoplasmic tail. And then we measured rescue. And by the way, we also did the same thing for GLUT2, the homolog. And what you can see here is that these minimal GLUTs rescue both the AMPA receptor responses and the NMDA receptor responses. So this means that GLUT1 and GLUT2 have the same functions and operate not by any conformational transmembrane region change, but simply by transducing an N-terminal cerebellum receptor, cerebellum response, cerebellum binding into an intercellular regulatory response that then controls AMPA and NMDA receptors. And this is schematically illustrated here, where you can see the 2 different neurexins as they bind to cerebellum 2, neurexin 3 and neurexin 1. They're binding to the same transduction machinery, then differentially regulates either AMPA receptors or NMDA receptors. And you might ask, how is it possible that this can work, because after all, this transduction module in the middle is identical for neurexin 1 and neurexin 3. We don't have a conclusive answer, but we believe that this is mediated by differences in affinities that neurexin 1 and neurexin 3 have for cerebellin 2. And thereby, they basically regulate the density of the responses on this GLUD that then differentially transduces one or the other signal. This is just one function of neurexin signaling. I've told you just one particular project, one particular story. Let me tell you, this is important, trust me, because NMDA and AMPA receptor responses are the two flavors of excitatory synaptic transmission. They have completely different downstream effects. They're operated in all new synaptic transmission, not just in plasticity, every time. And their relative activity determines the properties of many synapses and thus of circuits. However, neurexins have many other functions that I haven't had time to discuss with you. And the way I think about neurexins is as a Swiss Army knife, as a platform that does many different things. It recruits calcium channels presynaptically. It does all kinds of presynaptic regulations. It does postsynaptic AMPA and NMDA receptors. It actually regulates LTP as well, GABA receptors, endocannabinoid synthesis, what have you. It's all there. Various neurexin isoforms specify synapse properties via competing ligand interactions. They are the master regulators of synapse properties. So why should you care? Why is this important? If you are not interested in how circuits compute information, which you may very well not be, but you are interested in disease, let me tell you that this is actually very important for understanding both neurodevelopmental and neurodegenerative disorders. This really determines cognitive properties of a brain. And I will give you one example. What you see here is a map of the genetic landscape of schizophrenia taken from a recent paper, as you can see here. And what you can see is that there are some mutations, there are some genetic polymorphisms that are extremely abundant, very frequent, but have almost no effects, shown at the right lower side. And you can see on the left upper side that there are some mutations actually, which have huge effects, but are insanely rare, very, very rare. Importantly, neurexin 1 deletions are probably one of the most, if not the most frequent single gene deletions in many neurodevelopmental disorders, including schizophrenia. It's relatively frequent, much more than the more rare ones on the left, and its effect size is huge compared to the more common polymorphisms on the right. And what you can also see is that this relates to both a role for AMPA receptors and for NMDA receptors, suggesting that there's a pathway in schizophrenia, a genetic pathway, that may very well be a biological one, which links neurexins to schizophrenia. And so with this, I would like to close. What I've tried to illustrate to you is just one project. It's a project that sort of exemplifies how we are trying to solve the issue, the big question of how synapses are actually made and specified. The work we've performed and that I told you about today was largely carried out by a single postdoctoral fellow, Ginia Dai, who now has her own lab at Mount Sinai in New York City. It was also helped by Justin Trotter, a senior postdoc in the lab, and by Jason Aoto, a previous postdoc who now has his lab at the University of Colorado. Thank you very much for listening. I'd be happy to address any questions that you might have.
Thank you, Prof Südhof. We invite Prof Lim to moderate the Q&A.
Well, good morning, ladies and gentlemen, and good evening, Tom. I'm truly delighted to see you today, Tom. It has been a couple of years since we last met during your talk in 2019 at NUS, which I also had the pleasure of moderating. Thank you for joining us here today, Tom, and thank you for once again, as always, your very splendid lecture. I already see participants queuing up, so maybe we could start with the physical attendees first. Please go ahead.
Thank you, Professor, for this insightful talk. So I am Neelkant Kundu. So I'm from India and working in Hong Kong. So I have an engineering background and I have a general question for you. So as a biologist, what are your thoughts on the recent advances in artificial intelligence and artificial neural networks? So will it be ever possible that these AI models can beat this natural human brain, and what are the unique characteristics of brain which may or may not be simulated by a quantum or like a computer? Thank you.
What a great question. Very far away from my area of expertise. However, from my experiences using machine learning and AI in various contexts, including in the drug industry, my impression is that this incredibly powerful approach is better than any human will ever be at identifying patterns, processing big data, sifting through vast amounts of information. However, it is also limited because you have to train the machine in order to do what you want it to do. It doesn't generate its own ideas. It can randomly generate ideas and test them, but the parameter space is infinite. And at some point, that won't work. Let me give you an example. Probably in my own field, the most spectacular success of AI has been AlphaFold, which enables prediction of protein structures. And it's really a major achievement. It is truly something that is very impressive. You can put in any sequence and it will spit out a structure. And that structure turns out to be right many times, but it also has limitations. It can only do that because there's already so many structures. It cannot predict structures which are not conforming to existing structures in some way or another. So if you have something even as simple as something as a new kind of fold, it doesn't work anymore. Moreover, it can only approximate it. Its resolution is too low, for example, to really predict high-level side chain interactions reliably. So in other words, yes, it is fantastic. AI is gonna change how we do science, but it's not gonna change innovation. It's not gonna create innovation. It's gonna potentially make it easier for us in science to discern patterns, to identify commonalities, to make conclusions from vast amounts of data. And how we make these conclusions, what we do with these data, will be very, very important. Let me give you another example, which actually is very important because that's something that many drug companies have learned the hard way. AI is only as good as the database that you use to feed it. When you, for example, analyze RNA-seq data using Go analysis, you get all these functional networks and so on that people show in papers. Most of the time they're wrong. And they're wrong because the data that are fed into these Go analyses are literature data that are not curated, and most stuff in the literature turns out to be not very reliable. And as a result, all these data, all these Go analyses are often, they have some right hits, but mostly they just basically misunderstand. So it's very important here too, that AI will only be good for things where the data are good. It will be great for face recognition. It will be great for pattern recognition. It will be great for RNA-seq data in terms of getting the searching for sequences. But to identify biology, I think its value will be very, very limited. It will be great for drug design, for basically designing potential molecules that bind to drug targets. It's already revolutionizing drug discovery, those kinds of things. Yeah, that for sure. But not for, let's say, innovation. I don't think it will work very well.
Thank you, Tom. There is a line of participants waiting to ask you some question. So let me alternate to this gentleman before I take on the online question, please.
Thanks, Professor, for the talk. My name is Siu Jianjin, and I'm from Malaysia, working as a postdoc in Taiwan. So I have two questions. The question one is that, is the expressions of neurexin affect the cell-cell interaction between the neuron and other glia cells, such as microglia that actively involved in the synaptic pruning? And my second question is that, do you think the human brain organoid is a good model to study neurexin? Let's say the postdoc in your lab want to use this model. What kind of evidence that he has to show you to convince you it is a good model?
Yeah, these again are great questions. I don't know how synaptic pruning works. I'm kind of waiting for more data on microglia. It's clear that microglia are important in synapse elimination sometimes, but I'm not sure that they are actually generally involved, for example, in developmental pruning. I have my doubts about that. So I know this is by now in textbooks, but I don't think the evidence is very strong. As regards to, what was the second question again? Sorry.
About using the human brain organoid.
Oh, those brain organoids. Brain organoids are really a fantastic innovation to study early development. In terms of circuits, not so great. Their maturity is less than those in 2D cultures. So the 3D cultures, brain organoids, they never, the synapses, synaptic connections, there is very, very little. The best that can be done is by transplanting them. My colleague, Sergei Paska, has published a recent Nature paper, which I thought was really terrific, that showed that you can get some degree of integration of transplanted organoids, and that may work potentially to study that, but not in isolated culture. You see, the problem is that the organoids, as they exist now, lack basically a blood supply. So, obviously, so as a result, they don't really grow beyond a certain point. They start necroticizing and become funky, and there's lots and lots of problems. And so you can make two different cultures, and you can show that one culture extends axons to the other one, but that's not a circuit. That's just basically trying to get around the problem of the fact that it's 3D, but it lacks vital components that the brain has. The brain is not just neurons and glia. It's neurons and glia and the blood supply. And the blood supply is huge in the brain. So I'm doubtful that for studying neural circuits, the organoids themselves are going to be a good model. I'm hopeful that organoids that are transplanted may become a good model, but even there, we're just in the beginning of the field, but that's really where I think the development has to go.
Yeah, thank you.
Okay, there's a flurry of interesting questions online. So let me take one first before circling back to the physical attendee, pardon me. So Tom, there's a question on, is there a potential that your research can eventually contribute to improving the plasticity of the brain for individuals with advanced age? Over to you.
I hope so.
Me too.
So just to get into this a little more detail, we, among others, are trying to identify synaptogenic factors that might be decreasing with aging, with the hope that that will enable a potential treatment in future whereby not just aging, but especially dementia can be at least partly alleviated by increasing synapse numbers, synaptic function as a way of enhancing cognitive performance. I don't know if that's going to work, but it is remarkable that in neurodegenerative diseases, synapses are being lost, and that may be the basis, at least it correlates the best with dementia. And there, if one could prevent that or slow this down, that would be better than an antibody to A beta, at least as far as we know now.
Great. Yeah, I, for one, am really hoping that I could age healthily, particularly when I'm a neuroscientist myself. So let's get back to the online participant. The physical attendee, please.
Hello. Thanks a lot for the talk. I just had a question about the locality of some of these results that you showed us. And in particular, I was wondering, say you have a postsynaptic neuron with two different types of presynaptic neurons that express different neurexins or different types of neurexins. Do they each see some kind of average response postsynaptically, or is the effect localized to the individual synapses?
It's a great question. We have tested 3 brain regions for this paradigm, 3 different synapses. We have not tested the question you asked, which is different synapses from different brain regions on the same postsynaptic cell, but my expectation would be that it's specific for each synapse because it's dependent on the interaction. And so that if you have different presynaptic neurexin variants, you have different postsynaptic responses. In the 3 different brain regions that we have tested, it was very interesting in that in the subiculum, that's what I showed you, the data. In the prefrontal cortex, which we just published in eLife in a very extensive paper a couple of months ago, we see only the NMDA receptor responses. We do not see the neurexin 3 AMPA receptor responses. I have no idea why, because neurexin 3 is there. But that's what we see, okay? So the NMDA receptor response is very strong, AMPA receptor responses, neurexin 3 is not. In the cerebellum, we see only the NMDA AMPA receptor responses, not parallel fiber synapses, not the NMDA receptor responses. That is easier to understand because the parallel fibers don't have any NMDA receptor responses. They lack NMDA receptors, so that's better to understand. And we see a very strong AMPA receptor response. That's where it stands. My hypothesis is that depending on the complement of pre- and postsynaptic ligands, GLUD1 or 2 has to be postsynaptic, neurexin 1 and 3, SS4 plus has to be presynaptic, we will have different responses. In addition, cerebellum needs to be there. However, what makes things a bit more complicated is that neurexins also have other ligands. And these other ligands could compete with the cerebellums. And in fact, we have already shown that in a different paper where we showed that that competition can be helpful in avoiding self-inhibition in the olfactory bulb, which is a study I haven't discussed today. So you really have a very complex system. This is not a simple system where multiple molecules constantly interact with each other, and depending on what concentrations, what splice variants there are, they will form predominant synapses, predominant complexes that then shape a given synapse.
Thank you.
Thank you. I really don't want to keep the 2 ladies standing here for too long. So perhaps why don't both of you ask the question to cut down a little bit of time, and I see whether if Tom could take both the question. Please, go ahead.
Hi, Professor. That was a lovely talk. So I'm currently a PhD student in Cambridge. So my background's immunology, so I don't know very much about neuroscience, but I was just curious as to whether you had anything to say about, for example, Parkinson's and Alzheimer's and dementia, these diseases, they have very different mechanisms of onset and progression, and it's not entirely clear as to why that's the case, or, whether there's a difference in how neural circuits are degraded specific to these diseases. So is there anything known about what neurexins do in these specific contexts, or is that still something that's an area of research?
Thank you. Please hold for a while, Tom. Let me invite the other lady to ask the question.
Yes, thank you, Professor. My question is, I'm very curious about this perennial debate between nature or nurture, because some people say that us humans have a built-in template inside our brain when we were born, and it helps us to build predictive models of the world. I want to ask you as a brain scientist, what might be your insight to this question, or if you have a clear answer to this?
Okay, Tom, two rather generic questions, one on Parkinson's, Alzheimer's, the other nature versus nurture. Go ahead.
So Parkinson's and Alzheimer's disease are devastating and fascinating disorders that nobody understands at this point. There's tremendous genetic evidence which is beautiful and has advanced the field enormously, but for many of the genes, we actually don't know the functions and we don't know how these diseases develop. There's been beautiful studies performed by others that show that in at least some patients, there are changes in the synaptic machinery, including neurexins, but whether they're causally involved seems unlikely. I think it's more as an execution of a disease process wherein they might be contributing. We are actually very interested in Alzheimer's disease in particular, and there are talks online that you can see where I've talked about our work there. So this is one of the reasons why I didn't talk about it here, because I thought you could easily access those talks online. And I can tell you that synapses are a key element of these diseases that we need to address, I think, in the field. But at this point, we really don't understand these diseases. And so stay tuned, hope for the best. Hopefully we can, the field, not we, many people, a lot of people who are really, really good scientists are trying to resolve these questions now. And I'm sure that some progress will be made. As regards the nature-nurture question, that is, again, is a very, very important question. There clearly is no yes or no answer in the sense that neither one of them alone explains anything, but both together operate in tandem. I think there's no doubt that experience shapes the brain. And there's no doubt that the environment, in particular the history of a person, has a tremendous influence on brain development and on brain function throughout life. For example, we know from work of others that cerebellums, which I talked about today, are very plastically expressed. Experience causes a dramatic change in the expression of these molecules of cerebellums, not of neurexins, not of GLUD probably, but certainly of cerebellums, suggesting that, for example, at least in some neurons, experience could shape the complement of NMDA and AMPA receptors at synapses. So I think that in this eternal debate about nature versus nurture, we need to resist the temptation to want to find a conclusive one-liner. It's all nature, it's all nurture. Instead, we need to see this as a collaboration between the genome and the environment that together create the outcome, which can be and should be, and hope, a fully functioning, beautiful brain.
Yeah, thanks a lot. There are a couple of more interesting questions online, including one that talks about whether synapses are the driver or otherwise perhaps representing compensatory mechanism for neurodegeneration. Unfortunately, the event organizer has told me that we absolutely has to stop right here now. So Tom, thank you very much once again for the splendid lecture and for your insightful answers to the queries. Ladies and gentlemen, please join me to thank Professor Thomas Südhof for the talk.
Thank you.